Hi, good morning, everybody. Good afternoon. So it's my turn to thank everybody and the organizers to give me the opportunity to show some data and essentially in front of famous immunologists and biologists that I'm not because I'm a physiologist. So we're going to move toward diabetes after obesity. And basically, the topic is to figure out what could be the mechanism linking gut microbiota and diabetes and try to discuss with you the idea of a new paradigm, which is called here bacterial translocation. So we're living now an epidemic. Everybody knows that there's that epidemic of diabetes right now coming up, and the incidence of diabetes is really increasing a lot. Why? Because the pharmacology treats essentially the consequence, meaning hyperglycemia and body weight gain or excessive weight, rather than the cause. And this is the overall setting of pharmaceutical agents that are now available to treat the disease, and still we cannot really treat diabetes correctly. On the top of that, one major issue is that behind diabetes or following up diabetes, cardiovascular secondary effects are major, causing a lot of death and costing a lot of money. So definitely one has to find a clue for that, and we have to think hard altogether about that. So as I just said, we are right now treating the consequence, but we need to treat the cause most likely to figure out a good solution. For that, we need a new paradigm. So let's envision those paradigms. So far, we used to summarize, when we teach, we used to summarize that metabolic disease is the consequence of nutritional and genetic influences. And now we all know that there's the metagenomic impact, which is important, and we have to thank again Frederick Beckhead and Gordon's group that they really changed the way we do see metabolic disease, and it basically changed the life of my lab as well. So that's quite important. And he presented that, so I'm going to skip the causal role of microbiota. I just would like to point out that metabolic disease, as Frederick shown as well, are associated with inflammation. That low-grade inflammation, you can see macrophages here stained by F480, for example, in an adipose tissue. And so the key story was to figure out what could be the origin of that inflammation, and obviously what kind of molecule could be linking microbiota and inflammation of metabolic disease that was suggested to be causal, at least as a triggering factor of the disease. So the most simple way was to address the highly inflammatory factor LPS. And you all know about that structure probably. I'm going to skip to that. The key point was to demonstrate in animal models and as in human, as Dushko showed us, the mice microbiota is not human microbiota. So we have to find something common between both. And LPS could have been a good hypothesis. So we are looking to work with Professor Jacques Amar, who is an epidemiologist, and who had a plasma tech where we could assess LPS. And so the epidemiologist has been separated patients who were eating more fat or more high carbohydrate and more protein, and we assessed the amount of LPS, and the statistics show the beta score, and showing that there was a shift to the right of the mean curve for the LPS amount only for the patient who were eating more fat, meaning that the more you eat fat, the more you have LPS, and those patients were healthy, not diabetic, neither obese. So they were just having in their blood a risk factor, and we could demonstrate that this risk factor basically was a triggering factor of metabolic disease. So I'm just going to summarize on that slide a very long story short, and we could demonstrate in LPS receptor knockout CD14 for mice, or in mice infused with continuous low rates of LPS, or training mice with antibiotic, probiotic, prebiotic strategy, playing around with microbiota and with the amount of LPS, we could indeed demonstrate that LPS is a triggering factor. It's one of the first events, suggesting that a change in microbiota will lead to increased LPS, as in the blood, for example, of humans, and that throughout these studies was demonstrated as a triggering factor of metabolic disease. But today we have to go further and to understand better what's going on. So if the overall thought was that bacterial fragment could initiate metabolic disease, therefore we could raise two hypotheses. The first one is that those bacterial fragments could be considered as predictors, and therefore we could generate biomarkers biomarkers to predict the occurrence of metabolic disease, to diagnose or to stratify. And if there are indeed causal factors, definitely we can envision some targets and find some targets. So based on that, we analyzed the bacterial DNA distribution just by a general PCR method for screening method. And we compared actually saying that if there's some bacterial fragments, we should be able to find some bacterial DNA in tissues. And we compared whatever was in the cecum of a mouse with the mesenteric adipose tissue and with the blood. And much to our surprise, we did find bacterial DNA in the adipose tissue and in the blood of mice. The relative proportions are here. Obviously, those are tiny amounts, but still a highly significant amount of bacterial DNA. So bacterial fragments are present in tissues, at least in the mice, and we'll see in human as well. So we said, okay, we could find a bacterial fragment, bacterial DNA in the blood of mice. Is that true in human? So we got lucky again to get our hands on a cohort called the DESIR cohort, which is roughly made of 5,000 patients, randomly picked in the centre of France, in the center of France. And out of these patients, 357 of those patients did become type 2 diabetic. They were healthy to begin with, and 9 years later, they become diabetic. So we quantified the blood glucose, the blood DNA concentration, bacterial DNA, at the beginning when they were still healthy without any risk factors. And we analyzed what we found. And so we separate the patients in quartile here according to different amount of bacterial DNA into their blood. And we could find a threshold just right here showing that once you reach that threshold of bacterial DNA into the blood, you were predicted to become type 2 diabetic. So again, these patients are not type 2 diabetic to begin with at the time when we did assess the bacterial DNA amount into their blood. And for those who became diabetic, we could define a threshold of the amount of bacterial DNA. So that was true for the prediction of diabetes 9 years later after the blood samples. So as Dusko said, you want to keep the stools in the different studies. You also want to keep DNA in the different studies, but that is most likely done most of the time. So we can go back to that and do the analysis. That was true as well for the prediction of abdominal obesity. Again, those patients were perfectly healthy and lean to start up with. And 9 years later, we found out that the more bacterial DNA you had at time 0, the more the chances you have to have abdominal obesity, which is a strong factor responsible for type 2 diabetes and obviously obesity. So, but this type of prediction concerns a low percentile of the risk. And we asked the question whether an increased amount of risk factors, so we would like to predict type 2 diabetes, but we would like to predict that in a population who is at risk, so who have some obesity, who has hypertension, quote-unquote, the metabolic syndrome, but no problem with glycemia. Because the key point is to treat the patient at risk, those who will become type 2 diabetic anytime soon and who will develop cardiovascular disease. So we considered patient at risk. And we compared the most important predictor, which is fasted blood glucose, meaning that if you have a tiny bit of your blood glucose levels just slightly higher than the healthy concentration, you are really at risk. So that's the gold standard. And we compare this gold standard with the bacterial DNA and find out whether we could predict better. So the gold standard used to predict 10% of risk, which is high. You get 1 chance out of 10 if your blood glucose level is slightly increased to become type 2 diabetic. But when you have bacterial DNA into the blood, you can predict 15% of the population. So we can do better for the prediction of type 2 diabetes by quantifying the bacterial DNA just by comparing with the fasted glycemia. So that's just a general concept that tells us that indeed those bacterial fragments are indeed predictors of metabolic disease. Therefore, they could be cause as well and not only consequences. Here, they couldn't be consequence of type 2 diabetes because they were predicted before, they were assessed before into the blood. So for that, to identify which bacteria, to put a name of the bacteria, we use the the pyrosequencing method. So again, I'm not a microbiologist. I'm very far from bioinformatician, so I'm going to try to describe that. And basically, we took those two groups of patients. The patients, they were all healthy to start up with, but those who was supposed to remain healthy, and we compared to those who will become type 2 diabetic. And we pyrosequenced that, and this is the number of OTU that was found at 10% cutoff. So these are the different numbers of OTU find. You can find that it's kind of equally distributed according to the two population. And I have to keep that confidential for the sake of company, whatever it is, and so on. But you can find on here that when we analyze the different genera by bioinformatics, we could define that at least those two genera here represented something like 70% of the population. Therefore, into the blood, the diversity is way smaller, obviously, than one found into the cecum. Therefore, there's some kind of filter that tells that not all bacteria gets into the blood. A given amount of bacteria, which are represented here by those two major genera. And the second point was that we couldn't find significant differences in the distribution of those genera, whether you're bound to become type 2 diabetic or whether you were supposed to remain healthy. Therefore, that suggests that the mechanism which helped the bacteria to be into the blood is specific with regard to those which do not cross, but there's no difference between those two phenotypes. It's just a matter of the amount of those bacteria reaching, because the bacterial DNA amount into the blood is higher for those who will become type 2 diabetic. So which mechanism regulates that translocation, if we can call that translocation? I'll get back to that. How can we control that translocation of bacteria to the blood and to tissues, basically? So we used what people in infection, like Philippe and so on, have been qualified as bacterial translocation. I don't have to detail that in front of this audience. You'll figure that out easily, obviously. And so to study this mechanism in metabolic disease, we went back to mice studies. And for that, we've isolated an E. coli, a mouse E. coli. We labeled it with GFP and with ampicillin-resistant plasmid. And we just took mice, healthy and diabetic. We gave that E. coli into mice, and we looked 2 hours later where was going that bacteria. And so the point is that the first experiment that has been made was to scrap the intestine, the mucus, and to see who was present there. And we can see here the bacteria stuck into the mucus, and that was just from a diabetic mouse. And we compared that between healthy and diabetic mice and the different part of the intestine, duodenum, jejunum, and so on. And we quantify, that's quite easy. You just have to scrap the mucus, put it on a plate, and count the bacteria growing because they are ampicillin resistant. Or you can visualize that using the microscope. You can see only the diabetic mice are characterized by an increased bacterial adherence to the different part of the intestine. The key factor is that on that experience, we compared controls with diabetic and pre-diabetic mice. And pre-diabetic means that they've been fed a high-fat diet to induce diabetes, but we've studied mice before they became type 2 diabetic. And we can see that very early on there's an increased bacterial adherence at the level of the mucosa of the different parts of the intestine. So it seems that it's a very early mechanism that takes place as soon as you start eating fat, which is going to make you type 2 diabetic. So an increased bacterial adherence. And so we looked more carefully, and Chantal Chabot in my lab has been doing incredible work. She looked more carefully into the villosities, compared the control versus the diabetic mice, and you can see the fluorescent bacteria adhere here and not in the control mice. And she will have to enlarge that, if not, we won't going to see anything. And she showed me that there was some bacteria 2 and 5 hours later that were crossing, translocated from the lumen to the basal membrane of the intestine. So those bacteria were really much getting through the intestine only when the mice were fed a high-fat diet and to become type 2 diabetes. This is the the quantification of the so-called translocation mechanism. So we tried to identify which cells was taking care of those bacteria, harvesting those bacteria from the lumen to carry them over toward the basal membrane of the intestine. And she colocalized those bacteria with CD11C antibody, so therefore labeling the dendritic cells. And you can see here that, the red tag corresponds to dendritic cells. They are about to take care of those E. coli here, and if we enlarge, we can see better there's a yellow spot, which most likely corresponds to bacteria and E. coli that has been phagocytosed by the dendritic cells. And this one is about to be, this one is most likely being phagocytosed by the dendritic cells. Therefore, this is an acute mechanism. After only a couple of weeks of high-fat diet, the dendritic cells gets to the luminal side of the intestine and start grabbing those bacteria which adhere to the intestinal epithelial layer. So those bacteria were going further because she analyzed in the mesenteric lymph node as well, and she could find that spot here and this spot here, showing that the dendritic cells have been gathering the bacteria and most likely carrying them over to the lymph nodes, and where probably they've met the lymphocytes, I think, and doing whatever they have to do. So basically, there's a translocation toward the tissue. So we quantified that into the blood, into the adipose tissue, in the mesenteric lymph node, and so on, by quantifying the 16S ribosomal DNA to give numbers on that. And we can show that, she could show that when compared control and the pre-diabetic mice, there's a strong accumulation of bacteria into the blood. And this was true for the E. coli that we just gave age to study the dynamic of the mechanism, as well as when we quantify the overall gram-negative bacteria, because we've been working on the LPS hypothesis. So we wonder whether that was true of this type of bacteria. So indeed, into the blood, we have a translocation mechanism which takes place very rapidly after only 1 week of high-fat diet. So she quantified that into the mesenteric adipose tissue. You know that the fat pads into the intestine, into the visceral side is important to predict type 2 diabetes and so on. So she quantified that in a pre-diabetic mice and showed that it was already increased in the adipose tissue for the E. coli and the overall LPS and gram-negative bacteria. That was most likely the same thing. Therefore, this suggests that the translocation of bacteria, which is full of antigens and LPS, very inflammatogenic, is an early mechanism that leads to, a step-by-step mechanism leads to type 2 diabetes. So whether that's true in human, that was quite important. So again, we got lucky to have fat from healthy patients with an overweight, and patients with obesity, but no type 2 diabetes, because we're working on risk factors. So we gathered fat and we extracted the stroma vascular fraction, which is rich in macrophages, dendritic cells, and lymphocytes, whatever cells from the immune system are present there, and we characterized the bacterial DNA. So again, I have to hide that, but this is what happened. We've done parasequencing on these fat cells, and when compared to the healthy, the overweight, and the obese patients, we found out that this red genus here is increasing with the obesity phenotype. So not all the bacteria are increasing in proportion, only a few cells, that red and that blue most likely, are increasing according to the phenotype. Therefore, that was the demonstration that indeed we have bacterial DNA in our fat, because that's in human. This is concentrated in the stroma vascular fraction of the adipose tissue, and it increases, at least this genus here and the blue one as well, most likely, they increases with the phenotype. So this is the guy here. And the next step, and the last question, was to identify which genes are responsible for the translocation of those bacteria and bacterial DNA to the tissue. So again, in a mouse, the full bacteria is translocated because you grab the mouse tissue, you put it on a plate 2 hours after the gavage with the E. coli, and it grows. So therefore, the bacteria can reach the tissue. So we used LPS receptor knockout mice, CD14 knockout mice, and we quantified the DNA. And in absence of LPS receptor, the bacterial translocation is way lower. I mean, the amount of bacteria present in the tissue is way lower. And after we asked the question toward the intracellular receptors, so we got the mice from the Pasteur Institute, the not mice actually, and we could show that in the NOD1, essentially, but not the NOD2 mice, the bacterial translocation was reduced. Therefore, NOD1 is needed to induce the translocation, but NOD2 is not needed, neither in the adipose tissue nor into the blood. And as a consequence of that, we've analyzed glucose tolerance. So in this set of experiments, you administer glucose orally, and you feed the mice a high-fat diet to induce diabetes. The control mice, the wild-type mice, do become diabetic with hyperglycemia versus the mice fed the normal chow. But the NOD1 knockout mice never became type 2 diabetic. So therefore, indeed, in absence of NOD, there's a lowering of bacterial translocation, and the mice do reduce the induction of type 2 diabetes. And the NOD2 mice, they did become type 2 diabetic like the others. So basically, I was not involved as well. A major point was that MID88 is something quite important that basically recapitulates a lot of the inflammatory process. So we said, if we don't have MID88, what happened? Well, meaning that we will prevent inflammation. And we're very much surprised that actually to find that in absence of inflammation, we thought we will have the same phenotype as the NOD knockout mice, and actually the mice did translocate much more. Therefore, inflammation is important to prevent translocation at some point. That still needs to be debated, and the mice were much more diabetic. So again, the more you translocate, the more you become type 2 diabetic, and it seems to be an inflammation-related mechanism that needs to be discussed, and I need the help from immunologists to figure that out. To come back to metabolism, there's one key hormone which is quite important in metabolism, which is leptin. Leptin controls food intake, body weight, and so on, but it also controls the immune system. So we've studied OB mice, and we could analyze that bacterial translocation here mimicked by the GFP E. coli was dramatically increased. This is in log, so it's like 100 times increased in the fat tissues of the OB mice. So in absence of leptin, this is quite dramatic. If we made the OB mice into the background of CD14 to lower inflammation and to prevent, we could reduce the translocation event here. And obviously, the CD14 knockout mice have very low translocation mechanism. So we could reduce the translocation of bacteria in the fat of the OB mice. And we previously published that it was associated with a reduction of the diabetic phenotype as well, which is shown here. And therefore, leptin regulates bacterial translocation, and this is again associated with the metabolic phenotype. So one of the reviewers asked a very smart question that made us working a lot. He said, okay, but leptin works everywhere on every cell. So can you show that it's something specific to the intestine or not? And for that, I got a feeling I forgot I may have forgotten the slides. So, and for that, basically, we used a probiotic which produces leptin, and that probiotic was administered every day, and we got that probiotic from INRA, friends of Dushko actually, who was producing leptin, and when you gavage with this probiotic, you can indeed control, partly at least, the metabolic phenotype and the translocation. So the overall summary is trying to show and convince you that feeding a high-fat diet will obviously change the microbiota, that we know. And those bacteria will produce, secrete, or being translocated somehow into what we call metafactors, and those metafactors, like LPS and the others, will trigger cells in the adipose tissue and strong fraction that will generate many changes in terms of metabolic disease and essentially induce hyperplasia of the adipose tissue and being considered a risk factor for metabolic disease. So with that, I'd like to thank the people who most likely done the work. Most of it has been done by Chantal Chabot for the bacterial translocation, and Patrice Cagné has been done the work for the LPS story. And I'd like to thank all the collaborators here who have helped us a lot to figure out this story and to understand how it goes. Thank you.